Okay. Yeah. So, so day 5 today, and I think this is the last day of our presentation and also that program we have. I really enjoyed all of you, very talented people here. Good questions. So far, I have received a very good feeling that everybody is interested in the new science, right? I mean, future will tell who is interested, but I think uh, interest is important. You know, there's no end to learning, and I'm still learning from so many things. So today's topic, I mean, so day 1 we give you some introduction. What is emerging, re-emerging? We talk about day 3, uh, what the BSL2, BSL3 pathogens, BSL4 pathogens. We went yesterday also some BSL4 pathogens. Also the general principles you handle those pathogens in a laboratory settings. So this is the principle of all emerging, re-emerging diseases. But today's topic, I'll be more focusing, the more important is the diagnostics, right? Uh, what are the different techniques people are using for the diagnostics? And then after that, after the break, we'll talk about the vaccines. So far, what kind of vaccines are available and what are the future directions on this? Okay, any question? I have not seen you before. Oh, you are here all this time? Oh, maybe different t-shirt, okay. What's your name? Nandish. Okay, nice to meet you. So why that diagnostics is important? It's written there, but any additional thing? So why is diagnostic important? Because it's, yes, diagnostic means identifications, right? So it's a clinical disease. Laboratory is different. Laboratory diagnostics is different, okay? Like I'm detecting some genes, some bacteria, some fungi, labs is different. But when it is an outbreak happens outside, it's totally different than the lab diagnosis. There are a lot of regulations there. You know, people have to believe, okay, the data you are getting, it's right or not. Then it goes to the medias and people say, okay, there is an outbreak. CDC have to confirm, WHO has to confirm, okay, this is a new kind of disease. Otherwise, you cannot say, oh, this is this. But you have to, there are a lot of rules and pathways there because that is a clinical disease. So every aspect of public health activities depend on being able to measure specific health outcome because based on that, the policy may change, right? If you say, no, this is outbreak, but it is not true, people will be chaosed, people will do so many things, the trade market, the share market will be up and down, people will be panicked. So because the public health, but the government of India or any governments, their health department will make a policy, okay, there is a disease, maybe we should do lockdown like COVID. Government will make a policy based on the diagnosis, okay, there is a COVID outbreak in Gujarat or West Bengal, lockdown should be implemented, right? So it is a bigger thing than you do in the laboratory diagnosis. How the distribution of the disease happening, whether it is mostly localized in West Bengal or it is also transmitted to Orissa or Tripura or other places. Impact of the program on the disease. So the policy, what policy I'll do, and if the policy is there, how it will impact on the disease. Can we be able to control it? Or we need more force or external help from other countries to define those things, right? What kind of measure? So it is a big thing. When it's a clinical settings, the diagnostics is very important in that aspect. We cannot make it like, oh, I find it. But it needs to be characterized. The policymaker will decide, okay, this is their outbreak and what to take the next step. So clinical diagnostics and quality control. Clinical diagnostics are more rigorous than diagnostic in research, okay? Because it, it needs to be, as I mentioned, like it needs to be more quality control. You cannot say there is outbreak of anthrax or salmonella cholera, people have to investigate. You have to send the sample to CDC. CDC will say, okay, the same thing happened when COVID outbreak happened. All the samples has been sent to the CDC. CDC lab approved it. Okay, this is the virus is circulating in India, and they say yes, it's COVID. Do the lockdown, do these things, and also they provided more resources. Like, okay, what primary should be needed? They send a sample. This will be positive control, negative control. Do in your lab. Their people came to India to train them. Okay, you can do also. You don't need to send us sample every time because it will take time and cost. You can set up in your country, do this test on all those things. High level up. So this is more important. High level up quality assurance and quality control. That's more important. Like when you do diagnostics. Even like when you give a blood test, like you are saying, Sushant, like he has a dengue, because it's a clinical sample. It's not like, so they went there and they do all this testing, probably maybe PCR, or if it is an early stage or later stage, maybe they have done IgM or IgG ELISA. And then based on that, they say, okay, Sushant has a dengue. So that's why they need quality assurance. And the lab who is doing those, they have those certifications. They have a quality certifications. They are reliable on this. Okay, I think every year those diagnostic lab, they have to certify what the assay they are doing is correct. Some kind of protocol they have. Okay. So there is a difference between diagnosis and diagnostics. Okay? Detection of a virus does not mean it is causing disease. It's a diagnosis, right? So diagnostics means the protocol you are using to say this disease is present. When you find out, okay, I have a cholera or I have a dengue, that is a diagnosis. Diagnostics means you just did one experiment. Okay, this is this. But along with the diagnostics, you have to see, okay, Sushant is showing these symptoms. He has a fever, he has this, he has other platelets count went down, he has some headache, those things. And also they are checking their virus response like platelet counts or IgG count, IgG quantifications. Past history, whether he has before, what was he's taking before, or past he has any other infection or not. So those things combined together, then say, okay, Sushant has a dengue. So the diagnostic is a test, but the diagnosis is the combination of the diagnostic plus other things. Okay? So that's the say, a laboratory test and results is a part of the picture that a clinician uses to diagnose a patient. So diagnostics takes of different things. So there is a direct test, which is virus detection, which takes time. Whole virus, it takes more time. Viral nucleic acid, you can do real-time PCR on all those. Viral proteins, you can do different antigens. And different indirect tests. So indirectly, we are measuring the antigens, whether it is IgM, whether it is IgA or IgG. You can do more literature search. Those are the IgA is mostly present in the saliva, in any kind of secretions. IgA is the major. But mostly in peripheral, you see IgG, IgM more than IgA. Cell virus culture. So we can do embryonated eggs culture, always mice when used, so they inject the virus. Because suppose I got some sample and you are saying, oh, I don't know, I want to see if there is any virus or not, or if the virus is there, what it is. So then you take an embryonated egg, inject those. There are different techniques, like based on different viruses, you have to inoculate different places. Like some you can include egg yolk, some allantoic fluid, and then the virus grow there. And then if virus grow, you can see, it is very old techniques, and then you see, okay, there is looks like cloudy and something, and then you break the eggs, collect those allantoic fluids or yolk sac, and then you because if the virus is there, it will be a lot of viruses, and then people purify it, or you can do electron microscopy, all those things. Cell culture, it is very used. Primary cell culture. Primary cell culture is when you don't know is the virus can grow there, right? You have a cell culture, I got some virus and I have no clue is the virus grow in the vero cell or any other continuous cell. So in that case, people do mostly primary cell culture. In the, primary cell culture means you have to find out where you got the virus. Some say, I got the tissue from a lymph node. Then make a primary culture of lymph node and put it there. Secondary culture is mostly immortalized. When they immortalized means it is called cell line. And same thing, the more you'll passage, it will be secondary, again passage, it will be tertiary. And if you passage multiple times longer, then it becomes a cell line. And it calls mostly cancerous cell line use that, and you can indefinitely subculture those. Subculture means you have a cells, you just, there are protocol there, like trypsin, trypsin and EDTA, you add it, because it is a monolayer. Once you add the trypsin and EDTA, the cells are all separated. So it becomes single cells. And then you put it in 2 different plates. And then again it becomes monolayer. And then after certain days, so then it will be the passage. It is the first passage. It's called first passage. Then it grows fast, your flask is full of cells. Then you take out again, add trypsin and EDTA. Then again all cells dissociate and it becomes single cells, and put another 2 flasks. So that will be the second passage. So that way you can passage multiple times for cell line, because they are cancerous cell. You can do indefinite. Primary culture, if you do that way, after certain passage, they die, they don't grow. Because they are not cancers, they cannot grow further. They change their morphology also. So that is the difference. Question? No. Okay. Observation of infection on virus cells. So basically, you are infecting the virus in different cell culture and measuring what changes happening to the cells. Always you keep a control which has no infection, and another you put an infection and see the difference. Okay? And I'll show you some which is called cytopathic effect. You can do immunological staining. I have shown yesterday with the Nipah virus organochip model, like you use the Nipah virus glycoprotein, and you can see those green colored cells. That is called immunological staining. Also do immunofluorescent assays, and also you can do electron microscopy to see those viruses. So just basically infect the cells, see there is any changes in the cell happening, how we can measure, look over under microscope, oh, there is changes. So something virus is there. To confirm the virus, do staining. If you have an antigen tagged with any fluorescent fluorochrome antibodies, put it there. If it gives light, oh, that is the virus, specific virus I can detect. Or go to electron microscopy. You can see the real virus there. So this is a schematic picture of how it looks. We have shown you the inclusion body. Anybody remember inclusion body? So you can see the inclusion body in the cytoplasm. You can see inside the nucleus. Or sometimes some virus has a both within the nucleus as well as the cytoplasm. Okay? So based on those inclusion bodies, it's a combination, it's a lot of viruses together and make inclusion bodies. So it can be, don't need to have electron microscopy. So just ordinary microscopy, you can see the inclusion bodies easily. Or you may have a cytopathic effect. Cytopathic, cyto means cells, pathic means cell has some loss of structure or something. Okay? Pathology of cells, cytopathic. So the cell may go lysis, means you can see the cells are breaking apart. Some cells are intact, but some cells are breaking apart. Or you can see all the cells aggregated together, which is called multinucleated cells. Or the transformation, because the cell was there, it become cancerous. So you can see so much cells now. I infected the cells with virus, and now so much cells present. That means the virus may be cancerous and it makes multiple cells. So the transformations. So this is an example you can see under microscope. It is easy to see under my normal microscope. The cell lysis happen. You can see nice cells, and then you can see all burst out. Some are floating in the media. You can see the cells are floating. And if you keep longer, you can see all the cells came out because the cells are dying. This is the inclusion bodies. You can see within the cells, you can see those kind of bodies. Here it's present in both nucleus as well as in the cytoplasm. So mostly in the cytoplasm, I have shown, I think day 2 or so, or day 1. Pox virus produces the cytoplasmic inclusion body, and herpes goes in nucleus. So this is the cytopathic effect. They become syncytia. So bigger cell, multinucleated cells, bigger cells. These are the single cells, and suddenly you saw, oh, there is all aggregated together. That means virus is doing something to the cells. So that is all you're talking diagnosis, right? So then you can confirm, oh, something is there. I got it. I put in the cell, and cellular morphology are changing. So it is definitely virus. Now the question will be coming, I was expecting from you, oh, how you know it is why it's not bacteria or something? Bacteria or bacteria will contaminate this cell. Bacteria will grow there because grow in the media, but it will be all yellow color. It is contaminated. Virus does not contaminate the media. The color will be okay. If you're like a pink color or reddish color, mostly the media's color it's all depend on pH. If the pH is between neutral or 7.5, it maintains that. What bacteria does, it goes to acidic because bacteria produces so much, it changes the pH of the media. So it will be acidic, so go like 2, 3, and the media color will be yellow. So before you go to more exam, just by seeing the media color, you can say, oh, there is a bacterial contamination. So whatever you put it there in the cell culture, it's not, maybe virus there, but mostly it's bacteria because it contaminated your whole culture. But if you don't see, and you saw like syncytium formation or multinucleated, then it's definitely a virus. People also do, before putting in the culture, they filter it to make sure the bacteria are gone, whatever is coming out. So there is different pore size filter you have to use. So all the bacteria will be eliminated. Normally people use like 70 micron. Some people use 40 micron. The 40 virus will pass, 40 micron virus, all virus will pass 40 micron. But 70 micron bacteria will be eliminated, then you can put in the cell. So you know there is no bacteria, and just put in virus and then check it. So this is the transformation. So this cell are very few. Now suddenly so many clumping together and lot of growth happening. So definitely that contamination, whatever inoculum you put in the cell, something oncogenic because it is producing like cancerous, transforming the cells, and a lot of cells are producing. You can also use advanced imaging techniques. So they are huge. I'm not going to details each one, but mostly light microscopy and cryo-electron microscopy is done. There are so many light electron microscopy is there, wide field microscopy, super resolutions, and mostly the confocal and all those comes here. So there are a lot of, we need actually 5 colors you can do confocal. It's very tedious. 5 different color fluorochrome you can use and identify which cell the virus got infected. You should have a virus protein marker which is labeled, and you can put other 4 different, bless you, 4 different other markers to define what cell those are. So this is another way to diagnose what virus it is. So what you'll do, infecting after cells, you can fix those cells. You saw syncytia, fix the cell, and then do the staining and see under microscope. And then say, oh, I saw Nipah glycoprotein is present there. So definitely Nipah virus there. And then you put other markers, different CDs, like CD4, those like T helper, CD8, maybe CD8 cells. So put different markers and see what cells that got infected. So that way you can confirm. Another is electron microscopy. It needs expertise on this. and you need a good facility on this and also a good budget to have it. So different way you can do it. Electron microscopy, this is more confirmatory assays to do the to find out the agents. So check out those document to read more. So another assay is doing plaque assay. We mentioned, like, yesterday, Nipah virus, we put it there and we saw plaques. So that is the plaque. So each like a dot, dot, dot. So this is actually, I don't see too much dot. It looks like some are here, there's some are here. But when you see like this kind of dot, that means each virus particle go to that cell and killing those cells, and it's spreading. So if you keep it longer, I don't know which day it is, it's just an example, it becomes bigger and bigger, bigger. So that means the virus is replicating more, and it's killing all the surround cells there. Because the way it is done, you have the plate, you have the cells, put the virus, keep some time, absorption, because virus and cell needs some time to binding to the receptor, right? After that, you wash the plate and put an agarose layer so that there is no virus cannot go other side like this. So the viruses stay there and stays. That's why you cover it, kind of seal it with agarose, and then put in the incubator. Agarose also add also agarose some media so that it will also nourish those cells. The cell will not die. And then observe it. And you can see after maybe 5-6 days, you can see like this. And then you can count 1, 2, 3, 4, 5, 6, 7, that way. And then say, okay, I have 20 plaque formic unit from this plate. And then you have to calculate how much inoculum you put. So you do back calculation and then come out some number. Okay, I found 200 plaque forming unit per microliter of the inoculum. Okay?
So what does the plaque actually contain? Is it cells?
It's the virus infecting those cells, and so it's clearing up, right?
So those cells are lysed inside?
Lysed inside. Those cells are lysed inside, and that's why you find a hollow. And when you stain it, because it is, after all, like 5 days of culture, you stain those cells. That's why it's a crystal blue. After staining with the crystal blue, this area does not stain. That means there is no cell at all, because the virus already consumed it, killed those cells. And that's why you make the spots. Make it clear?
Yeah, what cells do you use?
This is mostly Vero cells. Vero, V-E-R-O. Yeah. It's an African grain kidney cell line. It's a continuous cell line. Also, you can do electron microscopy. Right? That's an example. As I mentioned, we talked about also the immunohistochemistry, the same like immunofluorescent or immunohistochemistry, all are same. Fluorescent means you are adding some probe which you can detect by laser, right? So it will give like red color, white, not white, red, blue, green, that different colors. You don't need here any fluorescent microscope. You can see under ordinary microscope. But here it's a different staining. It's mostly used like a diaminobenzidine staining, or HRP or horse radius peroxidase staining. So you can see like brown color. So that's very easy under light microscope. You can watch this. So here also the same thing. You add some primary, you infect those. These are infected cells, and then you add some maybe Nipah virus glycoprotein bind with biotin, not any fluorescent probe, simple biotin, because suppose you don't, in a lab you don't have a fluorescent microscope, you have only simple microscope, light microscope. Then I should add biotin, and streptavidin together, and you can see easily. But if I have a fancy confocal at least cost 0.5 million, based on what category and company you want. So it's very expensive. It's the expertise, but this does not need expertise, because that does not need those laser detections you can see under ordinary microscope. So that's But it has a limitation. You cannot do multiple colors and all those things. There are a huge list. It's a very busy slide. But the point is like protein to protein interaction, how it is happening. We can do different assays, right? Because virus infecting cells, okay? The syncytia formation happen. Why syncytia formation happens? Definitely there is some protein to protein interaction happening. As soon as I put the virus, within 2 days I saw it's a big clump of multinucleated cells. So definitely cell wall, another cell wall, all bind together somehow and they interacted and they make a bigger cells. So how we find out? So there are a lot of assays done, mass spectrometry, then affinity chromatography, all kind of chromatography, protein microarrays. What is changing happening? Earlier before the next generation sequencing came, people are using, we also use microarray. The genomic microarray you can do, protein microarray you can do. That's also the multiple probes used. Multiple protein probes used and put it there and see what protein is expressing. Basically, you are detecting what protein they have, okay? And then you say, okay, after virus infection, this protein increased because they interact with other and then the proteins. People do X-ray crystallography, again, expertise needed, NMR, spectroscopy, these are all fine-tuning, see, even give the structures of the proteins, how the protein folded and everything, more in depth of the characterization of the protein-to-protein interactions. A lot of study done how when the virus infects cells, what changes happening to the surface immediately. You have to do X-ray crystallography or NMR spectroscopy to find out that position, how the surface structure change, how the endosome formation, we talked about endosome formation and go to inside, how it happened, how the protein suddenly cell wall like this and make like a hollow inside and going there. So those are the study if you want to do, you have to follow this. And this is very sophisticated and they will give the whole structure as soon as you give the virus, what changes happening within the protein and the virus get inside. Probably you heard of Western blot. So the same thing, here is a cathode and anode. We always say like cat means cathode. It is always negative because cat in the light, that, right? In the night, the cat's eye looks like a line, right? In the daytime, it is not. So that's all as cathode means in the night, cat's eye looks like a negative, only line. So cathode is always negative. So if you put the protein here and just run the protein, those principles I'm not repeating. And then, so the protein separate based on the molecular weight. And then you just run it to transfer in the membrane. And then you do this probing to find out what protein it is, because protein already is separated. Because we use the SDS, trace glycine. You can do trace, you can do trace SDS. So it degenerate the proteins and make all those molecular weight different proteins and transfer it and detect if you have those. It's mostly used long ago before sophisticated technology came for HIV detections. For whether it is P24 antigen, it's a GAG protein or any protein, so they detect by this. And by that, they find out earlier, okay, that patient has a late infection or early infections, based on what protein they are seeing, early or late.
Is it true that the protein size that viruses have is much smaller than?
Yeah. You have to change the percentage of the jail what you are running. So yeah. So antigen detection. So exploiting the antigen and antibody complex. So how to do this? Produce an antibody specific for virus wanted and label it. So we do the fluorochrome. So check out the video. That's mostly we talked about to the ELISA. So by ELISA, do detect the antigens or you can detect the antibody. Okay. So there are different methods are there. You can do direct ELISA, indirect ELISA, sandwich ELISA, multiple different ELISAs there. So check out the video and then we'll talk maybe.
ELISA is short for enzyme-linked immunosorbent assay. With this plate-based technique, we can detect and quantify soluble substances such as proteins. During an ELISA, the antigen, which is the target molecule, is immobilized and specifically detected by antibodies, which are enzyme-coupled. When providing a colorless substrate, the enzyme produces a measurable, colorful product. An antibody is attached to the bottom of the well. This antibody is specifically designed for binding a specific protein of interest. Another soluble antibody is provided. The antibody is also specifically covering protein X. This antibody is conjugated to an enzyme. When we now add a substrate, the enzyme can convert it to a colorful product, which can be measured later. There are many variants of an ELISA. In the direct ELISA, the antigen is immobilized on the bottom of the plate, and the enzyme-linked antibody directly binds to the antigen. In the indirect ELISA, a primary antibody binds our antigen, and then a secondary antibody binds the primary antibody. The secondary antibody is enzyme-linked. The most common type is the sandwich ELISA. A primary antibody is immobilized and captures the antigen of a solution, and another primary antibody specifically binds the same antigen. But all three ELISAs have one thing in common. The last antibody binding is always conjugated to the enzyme which confirms binding. Here we provide an example of an ELISA. For this sandwich ELISA, we have two wells, both coated with primary antibody specific for protein X. On the left, we provide a sample containing our protein. On the right, we have the control without the target protein. Our antigen in orange will be immobilized. A washing step will discard everything else. Now we provide the enzyme-linked antibody. It will bind our antigen in the left well and remains bound after washing, whereas the antibody is discarded in the control. In the last step, a colorless substrate is given. Since the enzyme, in most cases horseradish peroxidase, is exclusively present in the left well, it produces a measurable, colorful product here. This can be investigated and also quantified using spectrophotometry. The control remains colorless since substrate is not converted here. If you want to have a closer look into the direct or the indirect ELISA, you may click one.
So got an idea? So that's why you detect antigen as well as antibody. So that method has been used for all, like lateral flow assays and direct immunofluorescent assays. You probably know the COVID when you have like take the nasal swab and put in this, and you can see those. So those are the lateral flow assays. The same principle here. What you do, like when you have a sample, right, the nasal swab, and say, okay, put in that tube and keep it 1 minute, and then put it here. So here, normally we have plate is already coated everything except this thing, right? Plate is coated. You have a control line, you have a test line. So this will be always positive. This and here is a combination. So those monoclonal antibody will be coated with colloidal gold, which will give color, right? Because we don't need a microscope or anything or spectrometry to detect those things. So it is you have to see by open eyes. So when these clinical samples happen, if it is positive, if I have a COVID or anything, and it is the specific one, it will bind to the antibody. Okay, it will bind to the antibody. So like here, it is the specific and bind to the antibody, and it is moving. That's why they say it's like keep for 5 minutes because it's moving. It's got there, it's moving, and the control bind here. You can see the control has a very strong line there, and if it is positive, it will bind there. If it is not positive, it will go away. So you don't see any line. So that's a nice technology. Lateral flow assay has been used for several diseases nowadays. But it has also limitations in the sense if those antigens either diluted or you are not, if you are in the first stage of infections, like maybe I got COVID, I'm only 1 day, so it is not come in the respiration yet. It's still, I'm in the incubation phase, so it will be negative. So that's why they say, watch. Maybe I have just fever, but it's still negative. But after a certain time, you can find then it will be very careful that you are very contagious at the point, because you should be separate from other family members, because you may transmit when it's secreting the virus, then it may contaminate others. So this is the same thing, like, just to show, it is the same thing people do use like a pregnancy test also. Same principle, it's a lateral flow. They are, pregnancy test, they use check the hormones, how much hormones you are secreting through the urine. Here for the viruses, they are checking how much virus you are secreting. Or sometimes they use mostly what antigens you are secreting from your nose or other secretions, and they detect it. So immunofluorescence is the same thing, it's same principle, what has been suggested, direct, indirect, or sandwich. But here mostly people don't do sandwich, it's a direct and indirect method. This is the direct means when you're detecting antigens, just remember this, and indirect always used for antibody detection. So fix as specimens in the tissues, incubate with the known antibody, because this is unknown, right? Antigen is unknown. I'm detecting antigen because I don't know. That's why you have to a known antibody. When you have a unknown antibody, you use known antigens, and that's the difference. And then detect undermost. So this is a same thing, schematic presentation, like you have a antiviral antibodies. You want to detect the antigens, you put it there, bind it. If there is no antigen, it will not bind it. Indirect, you are detecting antibody, same thing. You add the primary antibody, because I don't know we have the antibody or not. If it binds, then add another secondary antibody to detect, because it is a probe, and then you can see, yeah, that's the secondary antibody. If it is positive, if it already binds, if we see the antibody there, then it will bind, otherwise it will not bind, you don't see any colors. So it looks like this. So these are positive cells. You can see those cells are positive, and then here it's binding, but those not binding, any of them. So they are negative. So that means I can say if I am using for example, COVID, I don't know which slide it is, for spike antigens, right? And I have spike antigens, spike antibodies already there. I'm detecting antigens. So in that case, these cells may be infected with COVID, but I don't know. Then that's why I used antibody against the spike antigen, and that is labeled with this, and now this is detecting. That means this cell may be COVID infected, but not these other cells. And then what they do, if you do more quantitative, what area this is. And then they found out, okay, in that area, 1, 2, 3, that way they count the cells, and they say, okay, 10 cells per 1 millimeter square area, we saw with spike antigen positive. And like that, you have multiple slides, and you do like 20 of them, and then average of it, so which is more n values and more accurate, right? Maybe this slide you see more cells, the other slide may be very few, maybe another is more than these slides. So that way you make a nice standard deviations. The standard deviation, it's a curve. And so you cover the whole area and present value, so that is more authenticated than just reading one slides and report it. But the principle is against the same thing. You are doing the diagnosis of antigens. This is flow cytometry. It's very interesting. Here, this is the instrument. It's a schematic. This instrument is very big. So the cells are going here. So this is the cell injection tube. There is a seed fluid which is actually flowing all the time to make those cells very healthy and so that it can pass through. The laser is coming from here, exciting lasers, and there is a detector. So based on how many detectors are there, you can detect at least 40 different colors. It has a high throughput. It's a very highly sophisticated machines. And here what they are showing, the sorting, like, if I have a cells, I stand for 4 different color. Susan gave me a tube. Some cells, he's with different color, CD8, CD4, D163, or maybe 6 for for macrophage, 4 is for T helper cells, CD8 for cytotoxic. So give me a CD marker stained already and give me and say, hey, I need all cells to be separated from all those tubes. So this is the machine you can use. And here they will generate volts. There are plates there. And based on those voltage, and because those cells has a different antibody conjugated, right? It's already conjugated. That's why it's going through. And based on the voltage, you can change it and you can sort those cells. So one tube may be CD4, another state 163 or CD56, and all will be separated. And they say, hey, Susan, this is your cells. I separated everything. So that's the cool part of this instrument. You can identify what cell it is, as well as you can separate those cells, whatever you want. Mostly 4 colors, four-way sorting is, so far has been done. If you want to do multiple, then you can, there are a lot of ways you can do, you sort major population, then you stain again, sort again you take it out, stain again, sort again. So that way you can do multiple way of sortings. So this is kind of, you can see how so then you can confirm, my sorting is okay or not. How I can believe that machine is doing the right thing. So you can run it again and make sure those, okay, I saw the population only, I don't see other populations. So it will give like that kind of pictures. After sorting, once you run it, you can see, okay, I want this cell, so only this cell will be present, these are all be absent. But when I sort these cells, all will be absent, you can see only this cell. That way you can confirm, okay, what I sort is correct.
How do you have to purify the sample before you give it? For example, if I want to separate blood cells.
Oh, yeah, blood you cannot run. So you have to isolate the peripheral blood mononuclear cells. Okay? There are protocols there. Yeah, there is mostly they do the gradient centrifugation. So you put mostly the phycol or histopec, that's a kind of gradient. And then we know that lymphocyte has a gradient, which is like, I think 1.077, I forgot the unit. So that we know, okay? Mostly humans and animals, that is the lymphocyte density. You have to know those density. And then it's the market, you can give those density gradient. That means the solution. And put in the tube, and then put your cells. You can put the whole cell, whole blood also, and then spin down. So those cells, which is the specific gradient, it will be on that layer. The rest will go down.
So among that, this will separate.
And then you take that cells, wash it and everything, and then stain it. And then you give it to me or anybody who is doing this. Okay, can you separate it? Because it has only the lymphocytes, no other red blood cells and other things.
What about hard tissue? This has to be liquid, is it?
Hard tissue means what? Oh yeah, so you have to take the tissue, you have to digest it. Yeah, normally people use like either trypsin or pepsin, collagenase, based on what tissue it is. So you have to digest it, then the tissue will be separated, cells will come out, so it will be individual cells then. Then you do. Yeah. So yeah, it has a multiple application. You can do ion flux, how many different ions it's expressing. You can do cytokine analysis, what does different cytokines the cells expressing. You can do protein engineering. You can check what fold that protein it is. It's because you are detecting antigens. Immunophenotyping, we already mentioned. Cell viability, how many cells are alive, how many cells are dead, you can say. Microbial analysis, whether the cells are infected with virus or bacteria, you can just same thing, like tag it, and you can detect it as a positive and negative. So all the cell cycle again, very important, like whether the cells are in S1 phase or G1 phase, because that is the cycle name, S, G1, G3. So whether they are resting positions or the cells multiplication stage, whether the cells are proliferating, if they are proliferating, which stage those are, you can all define. Okay, and other markers too. So nucleic acid detections, I think what is that? Huh? No, biotechnology students should know those nucleic acid, right? So you have a PCR or? Yeah. So this is all about how to detect nucleic acids. Okay, so target amplification, I should not go too much on this. Basically PCR, tell me one person what this PCR is. Amplification of what? Your target gene, right? You're not doing the whole thing. So that if you want to do whole thing, then we have to do the sequencing to understand, right? But mostly the PCR is the targeted. Okay. Yeah. Yeah.
So I have a class till 12. I'll come after.
No problem. Yeah. So 15 minutes, right? 15 minutes. Okay. Thank you. So the PCR means it's a targeted gene, right? Target. I want to see whether COVID is there or not. I'm only focusing spike. No other. They have a lot of proteins there, a lot of genes there. So PCR always used for detection for this purpose only. But when you do, another talking multiplex. So because I don't know, Sushant gave me, always I'm referring to Sushant only, right? So I'll pick on another, you, right? What's your name? Haripriya, yeah. So Haripriya gave me a sample which has everything. He just want to fool me, and he put so many COVID, flu, everything gave it to me. And I'm confused what to do. And he said, okay, it has been so many there, tell me. Then I should use multiplex. Because then I am shooting probe against COVID, probe against flu, probe against HIV, whatever is there, and then run the PCR. And then I say, oh, these are positive for all this, negative for this. Then I can say, hey, this is the results. This is negative. So that is also used when a area you don't know what kind of pathogen is this, and you got a samples, so then you have to run those. Rather than running individually, you can run one experiments and find it out.
So they are very specific. So there is no possibility of a false positivity.
Oh, you have to standardize that. Initially, before you go that far, I have a multiplex PCR lab. You should be ready. You have a good control, you have a positive control, negative control. Your lab can do it. You have already tried multiple samples and you already proved you can detect. There is no, false positive, data is there. That's why you have to keep multiple controls there to make sure this is right.
Because both the infection have a similar structure or anything.
That's why you have to design. That's why you have to design probe nicely. You should not select a probe which has a security. No, no way. You have to see some probe which is totally different. That's why your primer should be, that's why you have to invest more time primer designing. Yeah, go blast search and see there is no homologies find out. It is totally clean. That's a long time investment. So another is nucleotide sequencing. You can do the whole genome sequencing. What's the advantage? I don't know. Oh, I'm so tired, primer designing those. Rather do the whole genome sequence. Uh, and BLAST will do this and tell me, okay, it is 99% homology, this organism, I'll do it. That's another way to do it. So, and another is microarray. That's another earlier, I think less used nowadays. It's a cheap format. It's very expensive though. I don't think a lot of people, if protein microarray people still doing, but genomic microarray is a little less now because people are going for sequencing. Because microarray has some also drawback. It has a false positivity. So you have different targets, right? And you mix those same kind of probes. And one chip may have like several thousand, targets. And you just probe and say, okay, detect, and same thing. So PCR, I'm not going here, maybe very brief. Okay, all the slides are coming. Very brief. If you want to do, we can talk later because we have another slide to finish, right? Already. So the same thing, like use the forward primer and negative primer, and you have low annealing, denaturation, and those things. They are the different temperature, and you do multiple copies, right? Everybody knows that, right? If you don't know, ask your friend, not me. This is very simple. You should know why. UG students should know this. Okay. So the next is sequencing. I like to give a little time, but not too much. It is the Sanger sequencing which earlier done, right? You should know that there are always some termination codons which cannot extend any further, and they have been mixed with all the nucleotides, okay? And there are some termination codons there. So it is just same thing. This template is forming with your RNA polymerase, and when the termination, it stops, right? And then you detect those. Either you detect by imaging, nowadays imaging, now, or you can take like people take X-ray plate or some, because they nowadays less people use the radioactives. We used long ago with the radioactives. It's also hazardous, right? We talked about yesterday. So that's why that's another way to do. But next gen sequencing is very good. Here with that capture, so it is in the plate, the capture, all those templates first bind. And then you have also those unextendable dyes, same like there, and also extendable bases. And it's bind, and when they detect, some of them are not binding, but some are binding unextended, so the laser can detect. And then it's been clipped. And then try to do extension, then it go extension. Then it's, as it is measuring, it's clipping, measuring, and clipping, measuring, and it's amplifying. And that way it's generating multiple images. And all those images combined together, and they say, okay, this ATGC or TGC also, and it reads all this. Then it gives sequencing. And it's very, very sensitive, very specific. Nowadays, has been a lot of, and there are a lot of companies making those NGS. This is recombinase polymers. What that does, it is also, this is all isothermal. What is isothermal means? Like a normal PCR, you have to change, right, temperature. You have to make the program. Here, just one temperature, okay? And that's good. So you need those things. Forward primer, reverse primers, recombinase enzyme. So they make complex. After making complex, what they do, there's a recombination happens. So the genes are changing, okay? And then they have been clipped, and then the polymerase try to bind. Then polymerase binds, so basically I'm coming here. So this was the initial, and this has happened. What changes happen? There is recombinations here, and that's happened. So it happening multiple times. And also another thing, after doing all those, you add fluorophores or quench. Quencher actually, it clipped later on. Yeah, it clipped later on. And then the quench, you add the fluoroprobe, so that means the same like fluorescent microscopy. You add the fluoroprobe, and then you clipped it, endonuclease, and then you extend it. And by polymerase, you extend it. So it is my target area, okay? And you extend it, and then it's ready. And then when you detect it, you can see the fluorophore and say, okay, this is positive. This is very sensitive and very specific, and this is the next generation of PCR, recombinase polymerase amplification. Another is the loop-mediated isothermal amplification. Again, it's isothermal, it does not need to be changing, but it needs multiple primers. Like, they are PCR only 2. Here maybe 4, at least 4, then above. And so because they have to make loop, like here, for example, okay, so here denaturation happening, and here, see that is a loop. You have to make a primer like this. It should not bind here if one see, but it will bind here. Okay, but the primer is another. So you have it. You see, the true primers are here. So one is making like this, and this also making like this. So then, same thing for the reverse also. At least 2 primers. You make this and this. So you make a lot of, so it's all the steps like this. Basically, you need more than 4 primers, and the idea is it should make a loop. And basically, you'll get that kind of structures. Because it is more sensitive. And it is making more copies of the same thing.
But how do you undo that structure after you're done with amplification?
What?
How do you undo that structure after you're done with amplification?
You don't need to undo it. It's amplifying, and then you run it and say, okay, this is there. Yeah, you can use the fluoroprobe also and detect it. Yeah, but basically, so PCR is very simple, and you need 2 primers, but there is chance of now the polymerase is more sensitive; it does not give wrong copies. But this is more, and the drawback of the PCR is you need to change the temperature often, right? But here there is no temperature change. It's very sensitive, and it makes a lot of loops. That way, you can detect the organism better. And it amplifies more than what PCR does. So that is the basic of isothermal loop mediated. And then another is CRISPR. So, hello. Tell him to play the video. So the CRISPR, I'll see the video first, and then we'll discuss. If you see the video, it will be more clear how the CRISPR diagnostic works. So basically, it needs a okay. It's the same technology, the CRISPR done for gene editing. Here is the same gene editing, but they're using probe. That I'll discuss once you see the videos.
In the corporate world, if you come across as too nice, people don't respect you more.
Yeah, after this video, yeah.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, that function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then, they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA-cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut and paste tool for DNA editing. In the future, scientists...
So you understand. It's a guided RNA and enzyme is tagged together. So you amplify by this recombinase polymerase or LAMP, right? So you have a lot of copies. Now you use those because you know, okay, I'm looking for COVID spike antigens, right? So you know the sequence of the spike for your target. And you already made this guided RNA for this. So you put this, all this in the complex, put it there, and it will cleave it. Once it cleave it, so there are one Cas12a, which is detector or Sherlock. And then those segment, then you put the fluoroprobe, okay? So now it is a probe, and then quencher will go out. Now we have the probe there. And that you can use. So this is specific for COVID spike now, because I know the sequence. My guided RNA already I designed. I amplified all those, like Shushant gave me the samples. I have a lot of RNA there, right? Now I have guided RNA, I put it there. It cleaves those area which are my site of interest. And then add the fluoroprobe, and then it is ready, and that is detection. And if it is binding with the fluoroprobe, that means, okay, that sample has the virus. If it is not binding, that means no, there is nothing, not specific for spike. Then you have to do others. Clear? Okay, maybe we should take a break, or you can come back.